Our white blood cells, those are the ones that are going to keep us safe. Yes? Fight this war for us. Now, what could possibly invade us? Any foreign cell, right? Bacteria, foreign cell. What if I put somebody else's foreign cells into you? Yeah, I don't want them there. They don't belong. They're invaders. What other nasty thing can make you sick? Viruses. Viruses have been around for a very, very long time. And chances are, when there are none of us left on this planet, they're still going to be around. Do you know why? They mutate very quickly, and they take advantage of other cellular mechanisms. Yeah, they're like little parasites. Yes? They take advantage of your cells. They take over your cells and cause your cells to make more of them. That's how they work. They actually can incorporate, and this is a scary thought, they can kind of hang out in your genetic material and hide there for a very long time. Did you have a question? Well, that's how HIV works, and we're going to talk a little bit about that guy today too. And mutations, mistakes, when they replicate, happen. And that's what's happened to the HIV virus as well. And those mutations can wreak havoc on somebody who finally figures out how to slow them down or stop them, because mutations can make them stronger, can't they? Just like bacteria. Bacteria have a very short generation time. Mutations are mistakes. All your cells do it. I mean, think, I have to replicate my whole genome every time I make a new cell. Am I going to make mistakes? Yeah. Some of those mistakes can be beneficial to the organism. Allow them to live longer in whatever environment they're in. Take your antibacterial everything that we have nowadays. Yeah. Maybe not such a good idea, all of this antibacterial stuff, because the ones that survive, that mutate, we kind of force them to reproduce themselves faster when we put them in a bad situation, which increases mutation rate. The chance that a mistake can happen that's going to be beneficial to that organism, resistance to whatever it is you're applying to them, is going to then select for them. That's called natural selection. Have you ever heard of that? Okay. So by applying all of these antibiotics and antibacterial things to bacteria, we might be causing more superbugs to appear on site. And we know we're doing that already. It's one of the reasons why every time you have a little ache, little pain, don't go and start taking antibiotics for it. Because most of the time, it's these guys. These nasty little viruses that are making you sick. And what can I give you to take care of a virus? Nothing. Who's going to take care of the virus? You. Okay, now there are some medications that can help block the virus's door in. And how does the virus get into your cell? You know, it's going to get into you, so it's in you, and it can get through the air, on surfaces, in your mouth, various ways. But how does it get inside your cells? You know all those little cellular markers you have, all those proteins you have we keep talking about on your plasma membrane? Special ones on your cells are actually the door in. It's kind of like the door they go through to get into your cells. Somebody mentioned HIV. HIV virus has a very specific door in. And we can find that door, and the protein's name is CD4. That's the door in for the HIV virus. And you know where I find the CD4 protein? What kind of white blood cells? Anybody know? T cells. In this chapter, we're going to talk about the extremely important role a T cell plays in a specific immune response. in adaptive immunity. So viruses like HIV use CD4, that protein on the surface of those T cells, to get in. And then what do? Well, they make their way to your nucleus, and they take over your DNA. And they cause your DNA, instead of transcription and translation for your cells, they take over the works and cause your DNA to make more of them. Now, once your cell has been invaded by a virus, it's a goner. But it can throw out a warning to other cells. And that warning, that chemical warning, is called interferons. And what is an interferon going to do? Shut those doors. It's going to interfere with the cell's door in. It's going to help block up those doors. It's kind of like locking the door. So that's once the virus is released, and it's going to be released, and in this diagram we can see the virus, that's that little cube-shaped thing, and the cellular marker, that's the little purple thing, and the interferon, which is released from the cell who's going to be a goner, but going to help this guy, protect this guy from being taken over. It's a big, huge realm of research to see how interferons can be used to help protect from viral infections. It's temporary enough so that the virus will die off because it can't reproduce itself anymore. Yeah, it's used as treatment for different viral infections, things like hepatitis. I think it's not permanent, but it's long enough temporary to thwart the virus from reproducing itself too much. That makes sense. We don't have anything that we can block that door yet. And that's one of the things that researchers, HIV researchers, are constantly trying to figure out. How can we block that cellular receptor so HIV can't get in? It's too smart. Once it's inside the cell, are you going to attack your own cells? Chances are probably not. As it progresses, your cellular markers may start to degrade, and then I might attack those cells, but not before that little sucker can start reproducing itself. So your body has to initiate something called a specific, or what's that other word called? We had innate immunity, non-specific, right? And what's the other kind of immunity? I'll give you a hint. It's called adaptive immunity. And that's a specific response to a specific antigen. All the guys that we've been talking about to this point are non-specific protection. They look at you, they know you don't belong, they're going to kill you. But adaptive immunity is specific. So interferons are antimicrobial proteins that are given off by cells to help protect cells from viral invasion. And there's a whole bunch of them. Sorry. Is there a list? There's a list somewhere. Oh, anyway, we'll get to it. So there's a bunch of different interferons, and they're viral specific. They're basically going to try to block the door in. Remember, T cells door in. What's the name of that door in for HIV virus? It's called CD4. We're going to revisit that again, so keep that in mind because that's going to be important. And it's why HIV is such a pain in the keister, because it's going to disrupt the whole specific immune response. The next type of antimicrobial protein is actually a group of many different proteins, and they're floating around in the circulatory system all the time. This group of proteins is called complement. And there's a whole bunch of them, 20 different proteins that, unless something's going on, they're just kind of floating around, hanging out, not doing anything specific in the circulatory system until they are activated. And it's kind of like a superhero when he gets that signal in the sky. Back to Batman again. What's going to happen when they are activated? And they're activated when a cell recognizes something foreign, eats it, and then actually tries to warn everybody else that somebody foreign is in the body. And how it does that is it takes what we call an antigenic determinant, and we're going to talk about that in a minute, and puts it out on the surface of the cell. That is also going to activate these guys to band together and poke holes in things. Guess what we call these proteins when they come together, band together, and form a structure that can actually poke holes in something like a bacteria. That group of proteins now is called a membrane attack complex. And complement will come together, ready for this, to elicit a MAC attack. It has nothing to do with McDonald's. Yes? MAC. MAC stands for membrane attack complex. And again, when we see foreign substances in the body, these membrane attack complexes will come together and form a structure that can poke holes in things. So remember that opsonization process? We said sprinkle some salt on, make tasty. These guys are involved in the make tasty part. So it's going to enhance the inflammatory response. It is going to promote opsonization, and that's going to enhance phagocytosis. And there's different pathways that will help with an immune response. So again, these membrane attack complexes are created by groups of proteins that band together called complement. And what's the name of the attack? Yeah, MAC attack, membrane attack complex. The other thing that can help reduce pathogens in the body is fever. So they talk about that here as well on page 775. Substances that are released, chemicals that are released from certain pathogens or from cells that are becoming damaged, called pyrogens, will actually cause your body to reset its thermostat. Now, what's your thermostat set at, average? 98.6 degrees Fahrenheit, 37 degrees Celsius, yes? But in the presence of these pyrogens, these chemicals, your body will actually up the temperature a tad. So these pyrogens, these chemicals, these pyro antigens, yes? Pyro means what? Fire. Will increase your core body temperature. That's what a fever is. You with me? Okay, so we have interferons. We have a MAC attack. We have some fever. That's going to hopefully get rid of those buggers. But if we still can't get rid of those buggers, we need to elicit an attack that's very specific to them. This is an example of an antigen, the red thing. And the whole antigen is typically large, but there's only certain parts of the antigen that our cells can recognize. Those are called antigenic determinants. And that's what a cell is going to target in an immune response. So if I was going to fight off a foreign invader, an antigen, I really wouldn't recognize the whole thing. I would only recognize what? The antigenic determinants. And this is where I use my sister-in-law as an example. You'll see what I mean. My sister-in-law's name is Jennifer. My kids call her Auntie Jen. Yes? All right. So if I saw Auntie Jen in a crowd, how would I recognize it was her? Probably her face, right? I'd say, oh yeah, there's her face. There she is, Auntie Jen. Keep that in mind for the rest of the discussion. So antigenic determinants is what our cells are trained to recognize. And that's what we're going to specifically fight in adaptive defenses. So adaptive defenses are specific. I'm going to see Auntie Jen. I'm going to see her face. I know she's the foreign invader. Yes? I'm going to fight that antigenic determinant. It's systemic because once I recognize that face and I produce chemicals that are specific against that face, they're always going to be present with me. And we'll talk about the cells that do that in a minute. And they have a memory. So the first time Auntie Jen comes to my house and I establish she is foreign, I will make cells that will never forget that face. So the next time she comes to my house, she's not going to make me sick because those memory cells will help get rid of her quick. Yes? All right, so it has a memory, it's specific, and it's systemic. Because those chemicals, and does anybody know what those chemicals are called? That I'm going to use to help neutralize foreign invaders? Antibodies. So in this case, I have an antigen, and this one happens to have 3 different antigenic determinants. So I'll have 3 different groups of cells that will recognize these specific determinants, A, B, C, and make neutralizing chemicals to neutralize them so other cells in an immune response can get rid of them. And in this case, 3 different antibodies, A, B, and C, are produced against this antigen. So back to antigen, remember, it's her face that I recognize. I can chop her head off, stick it on a pole, and show everybody else in an immune response. See, I enjoy this because I can show everybody else who's involved in immune response. That's what we're here to fight. If you see that, you attack. Okay? So who is it that does that for us? We're going to talk about a group of cells called antigen-presenting cells. And they are going to phagocytize antigen, cut her head off, and put it out on the surface. And show everybody this is who we need to fight. Yes? She was, I know, her ears must ring every time we hit the immune system. All right. So those are antigens. Antigenic determinants is what we are concerned with. Some antigens are what we call complete antigens. All on their own, they can elicit this immune response. But some of them are kind of like half antigens. We call those guys haptens. And you probably have come in contact with some haptens in your life. I'll give you an example of one that bugs the living daylights out of me every summer. Poison ivy. Is everybody in this room allergic to poison ivy? No. Some of you can roll around in the stuff and it's not going to bother you. How come? You don't get immune to it. In order for poison ivy, which is an oil on some plants, in order for that molecule to be a complete antigen, it's going to be locked onto with a specific protein. If you produce that specific protein that locks into that hapten, you're SOL, because you are going to be disturbed by poison ivy. And every time you become disturbed by poison ivy, guess what your body does? Makes more of those proteins. So you're going to be more reactive, and we'll talk about hypersensitivity in this chapter as well. If you don't make that specific protein, poison ivy is never going to be a complete antigen, and it's not going to bother you. It's not going to give you any troubles. Okay? So they talk about this in your textbook on page 776 when they talk about complete antigens and haptens. So complete antigens, they can elicit an immune response. Haptens, they're kind of half antigens. They need a protein carrier to elicit an immune response. So what's immunogenicity? Immunogenicity is the ability for something to generate a immune response. Exactly. So something that's immunogenic is something that has the ability to elicit an immune response and generate some of these neutralizing chemicals. What are those things called again? Antibodies. And some things are just reactive, and they have the ability to activate lymphocytes and antibodies and release chemicals like interferons, like activating complement. And those are what we call reactive substances. Okay? All right. Antigenic determinants described in the book. Let's talk a little bit about your self-antigens. So all cells, all living organisms, are made of cells with phospholipid bilayers and proteins dotted throughout. And everybody's a little bit different in the living cell world. Yes? Make different proteins, have different outer shells. Some have capsules around them. Some have cell walls around them. But the cell's a cell is a cell. Yes? And all cells have what we call self-antigens, or MHC. So in your book on page 777, they start talking about those self-antigens. MHC proteins. Your cells, when they go to T-cell kindergarten, are going to learn to recognize your proteins, your MHC, or self-antigens. Yes? Are your MHC proteins the same as anybody else in this room? No. So self-antigens are how our cells are going to determine who's who in the cellular world. MHC stands for Major Histocompatibility Complex. And there's two major subdivisions of MHC. MHC1 and MHC2. They're going to play a role in helping other cells recognize foreign substances that enter the body. So lymphocytes destined to become T cells have to go where to learn how to recognize your self-antigens? T cells. Thymus. Okay, so T cells are going to migrate through the circulatory system and go to the thymus. And you do most of this when you're very young. You've already taught many of your T cells what they need to know. And they're already in place, waiting for what? Foreign substances to come. Where are they waiting? They're waiting in lymphatic tissues, lymph nodes, tonsils, Peyer's patches. Yes? You with me? You've already done most of your schooling. Your thymus is kind of dinky and small. So if I want to see a big thymus, where am I going to see a big thymus? Yeah, in younger folks, until they've all gone to kindergarten, get put in place, and it doesn't have a huge role as we get older. When they go to learn self-antigens, it's called Immuno Competence. They learn what is self. What's going to happen to them if they flunk kindergarten? Apoptosis. What's apoptosis? Yeah, programmed cell death. We don't want you around if you can't recognize self-antigens, because what are you going to do? Attack my own cells. I don't want that. Yes, ma'am. Yeah, you could be fine without it. So Immuno Competence is talked about on page 777. When we talk about lymphocytes, there's two different types. Hopefully we figured that one out by now. What do we have? T cells and B cells. Where do the B cells go to become immunocompetent? Bone marrow. Their teaching will allow them to be what we call self-tolerant as well. So what's self-tolerance? Work well with self, right? Don't attack my cells. Leave my cells alone, right? And then we send them out into the surveillance areas. So they go from, if I'm a B cell, I'm then going to head out to a lymph node or a spleen or somewhere in the body. And if I'm a thymus, I'm going to be a T cell, and I'm going to go head out and park myself and wait. So the development of immunocompetence and self-tolerance is talked about in your textbook, and very important to pass the test so you can be allowed out into the rest of the world. If you don't pass the test, what's going to happen? Apoptosis. So, generation of antigen receptor diversity in lymphocytes and antigen-presenting cells is what they talk about next. And these cells are actually going to help show everybody in an immune response who they're after. And MHC1 and MHC2 are going to play a role in that. Before I get there, we're going to have what we call positive selection. T cells must recognize self-antigens in the major histocompatibility protein group. So what's going to happen, and there's several different scenarios here. Failure to recognize self-MHC, what's going to happen to my cell? You can see the little receptor deficiency there. There's something not quite right, you're dead. If you can recognize self-MHC, it's going to result in MHC restriction, which means I'm not going to attack anything that has what? I'm restricted. I can't touch those things with my MHC. Yes? Then negative selection is going to take place. So if you pass the test there, then you have to go to negative selection. So you must not recognize self-antigens after you've made that restriction or passed the restriction test. So if you do recognize self-antigens, fit in there together, what am I going to do to you now? So the negative selection is kind of the second test you have to pass. If you don't pass, bam, you're dead. Apoptosis. Failure to recognize self-antigens at this point means you have graduated T-cell kindergarten or B-cell kindergarten in the bone marrow. And where do you go? Then we put you out in the circulatory system and you take a trip to a lymph node somewhere. So that diagram on page 779 shows us T-cell education in the thymus. Oh, here we go with all these darn diagrams again. The next thing the book talks about is antigen-presenting cells. Those are the guys that are going to cut off the antigenic determinant, neutralize the nasty little critter that was carrying it, and show everybody what? Show everybody that antigenic determinant. In the case of my sister-in-law, cut off her head, put it outside, show everybody who she is. This is who we're here to fight. And that's what antigen-presenting cells are doing for the body. They are dendritic cells. They are some of the non-specific cells we talked about before. And they are going to help present foreign substances to the specific fighters in specific, or what's another name for specific immunity again? Adaptive immunity. Now, the next part of the book on page 780 is going to talk about humoral immune responses. Humoral. Where does that happen? Yeah, in your fluids. So, in this diagram, we see how we are going to create an army of fighters. And the army we're talking about in adaptive immunity, specific immunity, humoral immune responses are of two kinds. They're going to be the B cells, which are actually producers of those nasty little chemicals that are going to neutralize antigens. And what were those called again? Antibodies. Plasma cells is what we're going to end up with that make those antibodies. And the other very important player in this specific adaptive humoral immune response is the T cell. And the T cell is sort of like the cruise director of the whole thing. It's going to tell everybody what it needs to do, where it needs to go, who it needs to fight. So the T cell is the cruise director of the immune response. Now do you see why HIV is such a pain in the keister? If I inactivate the cruise director, everybody's going to be running out on deck and not know what the heck they're supposed to do. And people who are in end stages of HIV disease, who develop, what do they call that? AIDS, succumb to antigens that most of us just fight off on a daily basis because their cruise director is gone. Yes? They don't know what to do. They don't know how to fight. They don't know what to make. So they succumb to antigens, and one of the common ones is a lung infection. Well, it's called Kaposi sarcoma. You wouldn't succumb to that if your immune system was doing what it was supposed to do, if its cruise director was okay. So, adaptive defenses, humoral immunity. This is talking about the B cells. When B cells eventually come in contact with the antigen, and how might I come in contact with the antigen? It might bang into me in a lymph node, or my antigen presenting cells might say, yo, this is who we're fighting. They are going to start to change. They're going to start to make more of themselves, the ones that can recognize the antigen in question. You right now in your body have cells that will recognize any antigen you come in contact with, whether you come in contact with it or not, for the rest of your entire life. Because, yeah, but the cells will always be there. They have the ability to sort of switch up their genetic material a little bit. So you have cells that will recognize anything foreign you will ever come in contact with, ever. You can fight a war. Do you always win a war? No. And that's what we have to remember. So once the B cells have been activated, introduced to the antigen that in question, they are going to start to clone themselves. They're going to start to reproduce themselves. Some of them, with respect to B cells, will become plasma cells. And they'll sort of squeeze out of the circulatory system into the tissues, and guess what they're going to do for a living? They're going to make antibodies. Antibodies that will then circulate throughout the body. Remember, the antibodies are very specific chemicals that will help neutralize very specific antigens. They're also going to make what we call memory cells. So in the case of antigen coming to my house, I'm just one person. I answer the door. There she is. She's in. So I cut her head off, stick it out. Everybody make more. So now, when she comes back, I have centuries at every door. Is she going to be able to get in? It's right. We're going to cut her head off right away. So, memory cells. Memory cells. And I'm going to give you a not real example, but think of this and hopefully it'll make you understand. Say in every single lymph node you have one cell that can recognize whatever the antigen is. When you come in contact with that cell, you're going to go through this process. You're going to elicit an immune response. You're going to make plasma cells to neutralize eventually that antigen. And you're going to make memory cells. Now, instead of just one cell in every lymph node that can recognize that antigen, after an immune response, you've got 20 cells in every lymph node that can recognize that antigen. Yes? So the next time it comes along, and that's what the bottom part of the diagram is talking about, a secondary response, and that can be years later, we can fight the war a lot faster. We can neutralize whatever the foreign substance is a lot faster because I have a lot more cells that can fight the fight because of that initial response. So is that thing going to make me sick? No, because I can fight it off really quick. I can protect all the other cells from it because I have so many more cells protecting, surveillance, making antibodies, neutralizing. And that's what a secondary immune response is. So when we talk about clonal selection and differentiation of the B cells, this is what we're talking about. When they come in contact with the antigen, they're going to elicit a war, make a whole bunch of soldiers, and then those soldiers are going to be in place for the next time this antigen comes into our body. So say we have a virus that we've never, let's call the virus Sharon. That's my mother-in-law. You see a pattern? Yeah. So virus Sharon comes for the first time, makes me sick. Yes? I elicit a primary response, make a bunch of players, plasma cells, make my antibodies, neutralize Sharon, and have my memory cells in place. Next time Sharon comes to visit, she's not going to make me sick because I have a whole bunch more antibodies handy much faster to neutralize her before she can attack more cells. Yes? So, once we've gone through the process of an immune response, we have an immunological memory. And that memory is going to help fight off foreign substances faster by making more antibodies.
This is for
Every time. Yep. You got it. So, when that first response happens, and this is just a little diagram showing us antibody production from all the players that we just mentioned. And anybody ever have chicken pox? Yeah, in our day they used to make you all play together, so you get you would get the chicken pox. Okay, so day 0, exposure to the chicken pox virus. Yes, it takes you about 7 days to 10 days, right in here, to make enough what? Antibodies to do what? Neutralize the nasty virus. Unfortunately, you are infective between then. And you get all those nasty little pustules and you infect everybody else. Yes? So between 7 and 10 days, you're going to make enough antibodies to start neutralizing that virus. And you're going to recover, hopefully, eventually, from that nasty little invasion. Yes? What's going to happen the next time? Woo! The next time, the second exposure, maybe years from now, you get exposed to the chicken pox again. Again, this is a normal immune response. Does it always happen right? No. Normally, if your immune response happens properly, you're not going to get the chicken pox again. Right? Yeah, that's different. They can mutate and hang out in your nervous system. So if I get exposed to the chickenpox again, guess what happens? And it's not going to take 10 days for it to happen either. It's going to take, how much? 2 days? 1 day? I'm going to make how many more times antibody? Way more than I even need to neutralize the antigen in a much shorter time. How many times more, oh math people? This would be 10. That would be 100. That would be yes? I'm going to have 1,000 times more antibody than I even need in 2 days. Boom. Are you going to make me sick again? No way. Why do I have that? Because I have so many more cells now to make it. And how do they know to make it? Because they can recognize the antigenic determinant because of the memory cells I produced, which I have a lot more of now. Yes? Yes? So how can you get this immunity? Well, you can get sick. Yes? Or, so we have different ways to become immune or undergo this whole immune response thing. So humoral immunity can be achieved in 2 different ways, or actually 4 different ways. We can have active humoral immunity, where we cause our cells to go through this by exposing them to the antigen. Now, you can get that antigen naturally. Chickenpox come in, make me sick, get the little hives, do the whole thing. You get the picture, right? Or, what if I can cut off that antigenic determinant and stick it on something that's not going to make me sick? The cells don't know the difference. They see the antigenic determinant. They do their job. They're going to elicit an immune response. They're going to make memory cells. They're going to make plasma cells. Bam! Immune response. And guess what? I never got sick. I tricked my immune system into eliciting an immune response so I could have all those players on board just in case I ever do come in contact. And that's what a vaccine does, right? And that's dead. Doesn't matter. If I can recognize the antigenic determinant, I'm still going to elicit an immune response. Remember, that's the only thing that's going to fire me off. Antigen's head on a stick, right? Or attenuated pathogen. We're going to kind of knock out their genetics so they can't reproduce themselves. But remember, you're going to recognize them as foreign because of their what? Your antigenic determinants. Okay? We can also have what we call passive immunity. So, naturally acquired antibodies can be given to you to neutralize a foreign substance. So mothers can pass their antibodies to their fetuses. Sometimes if they have antibodies that will affect their fetuses adversely, that's not such a good thing. Remember when we talked about Rh positive and Rh? Yeah. So mother's milk is the best for that newborn baby. How come? Yeah, it has antibodies that can help neutralize any foreign substances that child might come in contact with in those first few months of life. Or we can just give you an injection with all the antibodies in it. That same discussion we had on the Rh positive and Rh negative mother. Anybody an Rh negative mother? What did you have to have when you gave birth? A shot of Rogam. What's Rogam? Yeah, that stuff, it's anti-Rh antibodies. So those antibodies, I can give them to you so they're circulating around in your circulatory system. They're going to neutralize the antigen. Your nonspecific cells are going to get rid of the trash, and you'll never do what? You're never going to elicit an immune response. So you're not going to have the war. the guy's in place, ready to attack. You see what I mean? You see the difference? If you're an Rh negative mother, they just do it automatically. It's safer that way. Yep. Every time. Okay? You understand why? Because if baby number 2 is Rh positive and the blood gets in, we're going to elicit an immune response. Baby number 3 is toast. Yes? Because those antibodies can pass through the placenta. So know the difference between active and passive immunity, and understand the difference between naturally occurring active immunity and artificially acquired active immunity, and then this stuff. Yes? And that's discussed on page 782 in your textbook. Now, the things that we are producing, these specific chemicals that we're producing to neutralize antigens, are called antibodies. And all antibodies have the same basic structure. That basic structure, and that's why you see all these little Y's everywhere in the book. You notice that? Why are they little Y's? Why aren't they little red dots or little green dots? Because antibodies have these same basic monomer structures. These are the building blocks of who? Antibodies. You're all in a coma. I'm so sorry. I'm really blowing your mind today. All right. So this is the basic antibody structure. All antibodies have a stem region. All antibodies have a hinge region. All antibodies have this little light chain on both sides. All antibodies have a heavy chain in the center. What's different about each of these antibodies? If they all have that, then why doesn't every antibody react with every antigen? Exactly. On the ends, we see what's called the antigen binding site. And that's what's going to be specifically produced when we make antibodies against a specific antigen. So the antigenic binding site is what is going to bind to the antigenic determinant. Yes? Say yes. Good. Now also in here, once these guys have plugged in, and it's kind of like Legos, when I plug them in, they're going to change shape a little bit. And that is going to trigger some other guys to do their job. Once an antigen has bound to an antibody, it's going to signal a lot of the different players in the war that we just talked about. There's different classes of antibody. And yes, you are going to have to know these too. Remember MAGE. M-A-D-G-E. Those are all the different classes of immunoglobulins. That's a fancy way of saying what? Antibodies. Ig immunoglobulin, IgM, is what we call a pentamer. So remember that little basic structure we saw before? How many? 5. How many antigen binding sites does IgM have? 10. See it? 1, 2, 3, 4. You get the point. Yes? So made of 5 of those monomers that all antibodies are made of. And there's 10 different places for binding of antigen to antibody. When I first get exposed to an antigen, I'm going to pump out a lot of IgM. Believe it or not, even though it's really big, it's not as efficient as a monomer antibody because it's too big. It's not going to be as efficient. But we make it first. So it's a potent agglutinating agent, and we're going to talk about that in a minute. The other type of antibody I make is something called IgA, and that's called a dimer. How come? 2 of the monomer units. Saliva, intestinal juices, mother's milk. They're going to help prevent foreign substances from invading. IgD, attached to the external surfaces of B cells. And this is how those B cells are going to know who's who. Because when those antigens come along, and did you notice on the B cell in the little diagram before, it had a whole bunch of little Y's on it? Guess what it was? Here, I'll show you again. Yeah? Okay. So IgD is going to help the B cells become activated so they can do what they're supposed to do during an immune response. The big player, the one that's going to help neutralize most of the antigens that come in, plasma cells are going to pump this out, IgG. IgG, immunoglobulin G, is a monomer, and it is the most abundant and most efficient antibody. So 75 to 85% of the circulating antibodies you have floating around in your circulatory system are IgG. So remember, I made IgG antibodies when I got the chicken pox the first time, right? And they're always going to be floating around in my circulatory system. You ever go to the hospital after you've had an immunization and they do something called a titer? What are they checking for? They're checking for how much antibody you have to that specific antigen. If your titer is low, was your immunization effective? No. What are they going to do to you? Zap you again, and hopefully you'll have a more efficient immune response, yes? So that's what a titer is. They're checking the amount of circulating antibodies you have to that antigen. And the higher that is, the safer you are, right? Because these guys are going to neutralize, bam, the minute somebody gets in. And if they're flying around the circulatory system, they're going to neutralize you before you even get a chance to make me sick. So IgG is the big player in an immune response in the neutralization of foreign substances. And then we also have something called IgE. A little bit larger, still a monomer. We're going to find it in places like the gastrointestinal tract, respiratory tract, tonsils, mast cells. Where do we find those? Connective tissue, very good. So we have constant surveillance in different forms throughout the body at all times. Are you going to have to know the different? Yes. Remember? MAG. So how do those antibodies work? All right. They work in several different ways. When an antibody and antigen connect, they form what we call an antigen-antibody complex. And think about somebody comes in my house, gonna rob me. I need the police to come. I don't have a gun. I can't shoot them myself. What could I do to neutralize them? You could knock them out or tie them up, right? What if I tie them up so they can't bother me and then I call the police to come get rid of them? Yes? That's kind of what an antigen-antibody complex is. The antibody is going to neutralize the antigen, not let it do its nastiness. So somebody else can come along and get rid of the trash. Okay? They don't kill, they neutralize. So, neutralization is going to help mask some of the dangerous parts. Neutralization can also tie them all up, bind them all up. When we did our blood typing, you saw that. That was called agglutination. Form what? Big clumps. So now all those cells can easily recognize the trash. It's all in big clumps. I'm going to go phagocytize it and get it out of here. Yes? Or it can cause it to come out of solution or precipitate. Who's going to come get rid of the trash? Yeah, phagocytes. So these complexes can actually create more phagocytes to come to the area and get rid of them. And during an immune response, some of the chemicals that are released can also activate, what's this little thing? Complement. What's complement going to do? These guys don't stand a chance. They have guys coming over eating them. They have guys all banding together and doing what? Poking holes in them. Yes? And the antigen-antibody complex is going to increase all of this activity in the body. Help to fix complement. Poke holes in thing. Leads to big holes in cell, guts spill all over the place. So you're going to make me sick? No. But I have to clean up the trash. So that's what antigen-antibody complexes do. All right, now here's the big kahuna of the immune response. So know what antigen-antibody complexes do. Understand antibody targets and functions. They are specific to their antigen. And because of their antigenic determinant, they can bind to their antigen. So that figure on page 785, figure 21.15, is your friend. Have you ever gone to the doctors and got one of those strep tests? And they go and they stick that swab down your mouth and make you gag, right? And then they go in the back room. They don't show you what they do because it's top secret. But what they do is they go take all the stuff they just got out of your throat and they put it on a little dish similar to what we did when we did the blood typing. Yes? And then they take antibodies that were made very specifically in other poor animals like bunnies to the antigens they're looking for. So when you go for a strep test, it's a very nasty little streptococci that causes all kinds of problems. It's beta hemolytic, sore throat, causes all sorts of nastiness. But it has very specific antigens on its surface. And somebody in some lab somewhere created antibodies to the antigens on that strep bacteria. And they harvested those antibodies and put them in a nice little tube for your doctor to use. You see where I'm going? So when the doctor takes the, yeah, the rapid strep. A lot of those rapid tests are the same basic principle. We take an antigen, we apply a known antibody to it. If we see a reaction, it's there. So back in the secret room, that's what your doctor is doing. He's taking that tube of that some poor bunny gave up its plasma for. And what's floating around in the bunny's plasma? All the antibodies that it made to that strep. He puts it on there. If antigen and antibody are both in the sample, they're going to get clumps, agglutination. That means you have a positive strep test, and he writes a nice little prescription for you and sends you on your way. Yes? Some of you say, well, hell, I've gone to the doctor before, and that test didn't work, and I ended up having strep. Why do you think? It was too early. Maybe there wasn't enough of that bacteria in the sample he took when he shoved that thing down your throat. So he didn't get a noticeable reaction. The best way to tell whether you have strep bacteria is to take that same swab and then rub it on a petri dish. Yes? So even if there's one or two of those suckers in there, guess what? They're going to grow up on the petri dish, and the person in the microbiology lab says, aha! This person has strep. Yes? So, they talk about something called the monoclonal antibody. And what I can do is elicit an immune response in something. And I used to torture many, many mice. I have tortured many mice in my lifetime to make monoclonal antibodies. What I do, or I did back in the day, was I injected a specific antigen into my mouse that was foreign to my mouse, that I wanted to make antibodies to. What happened to the mouse? Went through the immune response, created all of those memory cells. Where are all the memory cells in my poor sweet little mouse? Where are they going to hang out? In the lymphatic system, in their lymph nodes, in their spleen. And then we went to get the cells. How do we do that? It wasn't very nice, not for the mouse. And rip out their spleen. Highest concentration of those cells, pretty big lymphatic organ, right? And then I went to find them. How do you think I did that? Well, I brought the antigen with me and I coated it on a plate. And I took some of the antibodies that certain cells made and I applied it to my plate. If there was a reaction, that cell made the antibody I was interested in. What if I could just take that cell and cause it to make tons and tons more, just like it? Now what do I have? Tons and tons of antibody made from one cell, monoclonal antibody. Yes? So I caused that one cell to make a whole bunch more friends, and I was able to get tons and tons of antibody that I wanted. Now, here's the deal. I got to end class, but there's a few more pages left in this chapter, as you can see. All right? Your job is to go home and study this stuff. We're going to finish this on Tuesday. Are you going to have an exam on Thursday? Yes. So what I want you to look at, the reason I put this up here before you go, this is the whole spiel. All the players in adaptive immunity. We already recognized the B cells, but these guys are also going to help, and these are the T cells. We already talked about the CD4 positive T cell. That's the cruise director. There's also CD8 positive T cells, kind of a different receptor. They're both going to play a role in the immune response. Okay? So your job this weekend, don't be slacking off, because this is hard stuff. Finish this chapter. We'll finish it together on Tuesday. Okay? Quiz? Nah. Maybe we'll do clicker stuff. I know.